All right, welcome to photosynthesis. And what you see before you is a beautiful green, looks kind of like a temperate tropical temperate rainforest as opposed to a tropical one, maybe up in Seattle somewhere, but I don't really know. It could be anywhere. But it's a beautiful, lush green environment, and what we're going to be learning about is how all that green works, how all those plants, how those autotrophs work to do some photosynthesis. It's an amazing thing, and without it, you and I would not be here. So, photosynthesis, making life from light, air, and water. It's not quite that simple, but it's astonishingly, if you break it down fundamentally, close to that. So, all life, before we jump into exactly the nitty-gritty of photosynthesis, we need to kind of understand photosynthesis place in the grand scheme of things, of life on Earth. And in order to do that, we need to think about the organisms that do it, the autotrophs, and how they compare with heterotrophs, okay? All life, whether you are an autotroph, a plant, algae, photosynthetic, anything, or even a chemosynthetic organism, all life, autotroph or heterotroph, needs a constant input of energy from the environment. The minute you stop taking in energy is when you are unable to maintain homeostasis and you are dead, okay? Now, heterotrophs get that energy by eating food. They eat organic molecules, okay? And because of that, remember, we call them consumers. Autotrophs need food also. They differ from us heterotrophs because they can make their own food. They do not need to go out and kill something and eat it, go to the refrigerator and make a sandwich. They make their own food right in their own tissues. And because of that, we call them producers. Now, the way the organisms that we're focusing on in this unit do that is they convert the energy of sunlight into food molecules, okay? They use the energy of that sunlight to build the complex organic molecules that they then will break down as food particles. That's photosynthesis, building organic molecules using carbon dioxide as the fundamental way to get the carbon. Remember, organic molecules require the presence of carbon. They're going to use the CO2 and the energy of sunlight to push those CO2 molecules together into a big complex molecule, and then they're going to break it down and get energy out of it. Now, we convert the food molecules we eat to energy by way of respiration. They do the same thing. They convert their food that they have in their cells to usable energy by way of respiration. The only difference between heterotrophs and autotrophs, between autotrophs and us, is that they can make their own food. We can't. Either way, the food that is either obtained from the environment or made in their tissues has to be broken down and used to make ATP. So, please understand that if on a test of any kind, we don't start thinking that only animals need to do cell respiration because plants do photosynthesis. That is not true. Plants do both photosynthesis and cell respiration. We can only do cell respiration. Okay? Now, so heterotrophs and autotrophs both are doing this. Okay? I hope you recognize that. If you don't, come to me tomorrow and I'll smack you. All right? This, of course, is cell respiration. We are going to oxidize this glucose molecule, meaning strip electrons from it, and we are going to give them to oxygen, and we're going to be left with poor little CO2 molecules, but in the process, that energy from those electrons is going to be used to make ATP. Okay, this is an exergonic reaction. We've talked about that in class. Exergonic reactions release energy, and this right here is the energy that is released. Okay, now autotrophs in their photosynthetic reactions, remember autotrophs have to do this too, but before they do that, they will make their own food. And this is how they do it. The exact opposite, okay? Instead of oxidizing that and getting CO2, they are going to take CO2 and they're going to reduce it. They're going to add electrons to it. How are they going to do that? Well, they're going to use some light energy to help them do that. They are going to add electrons to this, protons as well. Remember, electrons travel with protons, and they're going to reduce it, and by doing so, they're going to make this big, enormous, complex molecule, okay? They're also going to give off some of that as kind of a byproduct, okay? So this is reduction here. It is an endergonic, energy-storing reaction. This is energy releasing. This is energy storing. We are storing energy in that. Remember, ATP is lousy at storing energy. It lasts only a few seconds before it's degraded and used up. It's very unstable. This glucose molecule, on the other hand, is very stable. It's a good way to store energy. Okay. All right. Please be aware, we've talked about this in class as well, that plants are not the only autotrophs. There are bacteria that can do photosynthesis, just dandy, cyanobacteria, that blue-green bacteria. So many protists, the algae, there's some sea lettuce that you'd find easily down at the Chesapeake Bay, doing photosynthesis all over the place. And of course, plants do photosynthesis as well. So if you are a plant and you're going to do photosynthesis, you need light energy. You are going to use that light energy to make a very temporary energy molecule that you will recognize, ATP, and then you're going to use that ATP to build glucose, okay? So we don't usually, when we see that photosynthetic equation, that CO2 plus H2O plus light energy is going to give us glucose plus oxygen. I don't see ATP anywhere in that setup, okay? Because this is kind of just an intermediary, okay? The light energy is going to be used to make this, but it's going to be spent very quickly to make that. So what we focus on is the light that goes in and the glucose that comes out, and we will see how this intermediary is used, okay? Now, in order to make that glucose, the ATP energy is going to work with water and CO2 to make that glucose. So this is essentially what the glucose will be made from, are those things. Okay. All right. It helps to know a little bit about plants if we're going to talk about photosynthesis. So please understand that plants' bodies are designed to obtain the things they need, like sunlight. Of course, they have these broad leaves that are basically solar collectors being held up to the sun, full of their photosynthetic pigment to catch sunlight. CO2 enters plants through their stomata, which are holes in the underside of the leaf that the gas can come in so they can get their CO2 they need. Water is going to be obtained from the roots, and the roots are very cleverly designed with little tiny root hairs that increase their surface area. Very often there are fungus living in symbiotic relationship, a mutualistic relationship with plant roots, and we call that mycorrhizae, and the fungus get all wrapped around the roots and stretch out into the soil even further to get more water. So they have very clever strategies to get lots of surface area, to get lots of water. And then nutrients, things that are in addition to CO2 and water, but that they still need. Potassium, sulfur, magnesium is really important in making chlorophyll. All these things, they take that in from the soil as well, in the roots. Now, leaf structure is really, really important. Mesophyll is the middle of the leaf. In class today, we talked about the cuticle, which is the waxy covering, the upper epidermis, which is a single layer of cells, and then there's this lower epidermis, and you can see the holes where the CO2 will be allowed to come into the leaf. But the middle part is where the photosynthetic action is, and in particular, this upper middle called the palisade layer, okay? The palisade layer is where the lion's share of photosynthesis occurs. These cells are chock full of chloroplasts. They are just teeming with chloroplasts, okay? The spongy layer down here, this lower part, there's chloroplasts in these cells too, but what's more interesting here is all the air spaces, and that's so that CO2 can come in and we can have lots of CO2 coming in here to feed the photosynthetic activity going on up here in the palisade layer, okay? The lower epidermis, again, I already mentioned, but this is a nice, really super electron micrograph of a stomata. This is the hole through which the CO2 would go in. And these two kind of brownish things on either side are guard cells, which allow that hole to open and close. Because remember, we don't want to lose too much water. Plants are land organisms, but if they lose too much water, you know what happens to a plant. I kill them all the time by not watering them enough. They will die. So these stomata can close in times of drought to try to help the plant keep water in its tissues. Of course, the bad thing is if the stomata closes, of course, guess what? You can't get in. No CO2. So then you have the problem of not being able to conduct photosynthesis properly. Okay? So the stomata is very, very important in conducting what is called transpiration. Transpiration is how the plant gets water into its roots all the way up, if you're an oak tree, up that giant plant body and out through the leaves. So we'll talk more about that later, but it's also, of course, obviously important in gas exchange. So stomata play important roles in getting oxygen and CO2 in and out of a plant, but also in getting water all the way up a plant. We'll talk a lot about that later. All right, we need to do some chloroplast anatomy. So imagine those palisade cells, plant cells, full of chloroplasts, little organelles. Also, the cells would have a nucleus and all those other things we associate with cells, but we're going to talk about this chloroplast. Okay, so leaf, we're going to zoom in. Right here would perhaps be a palisade cell, or maybe it looks like actually they're doing a spongy layer cell here. And then we're going to zoom in on just one of these chloroplasts, and there he is. Now, if you will look, they've done an electron micrograph down here so you can super detail in real life. This critter has two membranes, just like somebody we just visited. You remember our mitochondrion has two membranes? Well, this guy has two membranes, an outer and an inner. And again, this outer and inner membrane indicate a probable endosymbiotic origin for this guy. It was probably originally a free-living bacterium that was photosynthetic and got engulfed. Also, chloroplasts do many of the things mitochondria do, in that they can divide and make new chloroplasts, and they also have their own DNA. All right, so what we need to focus on, we got an outer membrane and inner membrane, and we've got stacks of these little structures inside the chloroplast, okay? These little stacks, they look like stacks of coins, okay? A stack is called a granum, but you will probably not use that word very often, okay? What word you will use is the word for each individual coin, and that is a I'm going to run out of space. Let me write it up here. Thylakoid. Each little coin in the stack is a thylakoid. And the space that surrounds all the thylakoids is fluid-filled, and it is called the stroma. The stroma. It's fluid-filled space. You really need to understand about the thylakoids and the stroma. Everything else is kind of inconsequential, other than it's inside a chloroplast, okay? All right. We'll talk more about that. All right. So here's some better pictures, but what I just said, this is nothing new. Stroma is the fluid-filled interior, stacks of thylakoids that are surrounded by the stroma. The thylakoid membrane, so here we are, this is a thylakoid membrane right here. The whole thing is a thylakoid, but the membrane around the thylakoid is really important because that's where the chlorophyll is. All of those structures, the chlorophylls are little molecules embedded in the membranes here, okay? So that's where the light trapping occurs. All right. Chlorophyll, an amazing, amazing molecule, and I'm having trouble. Okay? Please understand, without this, life as we know it would not exist. Chlorophyll is what allows us to harness the energy of that beautiful, beautiful sun out there and use it. If the plants could not do that, we would be in huge, huge trouble. Okay? All right. Chlorophyll looks like this. It's a big complex looking molecule, and it's got a little magnesium atom. Remember, that's something they have to get from the ground, a little magnesium atom under the ground, from the soil, and they embed that in the center of a chlorophyll molecule, and again absorbs light energy that the plants use. Now, some basics about light. Remember that light behaves both as a wave and as a particle, but in biology, we are going to talk about its particle nature, and we're going to talk about light in units of photons, okay? It's a fixed quantity of energy, okay? Now, chlorophyll doesn't just absorb any old light colors. It is very best at absorbing red wavelengths and blue wavelengths, and it is terrible at absorbing green, which is why when we look at chlorophyll in leaves, we see green. It is being reflected back. It is not being absorbed. So the spectrum of light that's doing a plant good is the red and blue ends, not the green. That's useless to a plant, okay? So it reflects green. It does not absorb it, okay? Now, this is showing, of course, the Roy G. Biv spectrum, red to violet, okay? And what this is showing is the peak absorption of chlorophyll, okay? And you can see, I'm not sure what these different lines are indicating, but the two peaks for chlorophyll are here in the blue end and here in the red end, okay? Of the spectrum. All right, we know, because you all separated some pigments, that there are other pigments besides chlorophyll A. Chlorophyll A is the important one. It is participating directly in the reactions that are going to do photosynthesis, but there are other pigments as well. Chlorophyll B is more of a yellow-green, perhaps seeing some in that. Those might be in actuality more carotenoids though. And there's also carotenoids, which we think of as more in the yellows and oranges and reds, okay? Both of these other accessory pigments can help to catch some light, but mostly they are in a photoprotective role to absorb and dissipate damaging UV types of rays and so on, so the plant cells don't get damaged. Okay? All right, let me see if this is anything we really need. I don't think so. This is just showing you what we already did. Show you a little bit earlier. Chlorophyll A is best here and here, as is chlorophyll B here and here, and even the carotenoids are better up in this blue end. Okay, so just remember, we're going to be using chlorophyll A is the really important one, but all of them do better in the blue end or in the red end, and they're rotten at absorption here in the middle. Okay. All right. One last thing. Very important to remember before we move on to the rest of our little photosynthesis talk. When chlorophyll is struck by light of the appropriate wavelength, electrons are excited. They hit that photon of energy, excites them, and they boost to a higher energy level. Now, normally, electrons get boosted to a higher energy level, they fall back down, and they release light. as part of that release of energy as they fall back down to their ground state, their unexcited state. But in photosynthesis, the electrons get excited and they get caught before they can fall back down. That's really important, because if they didn't get caught, they'd just fall back down, release a little light, and we wouldn't be able to use that energy. But they get caught, and that energy gets harnessed to make sugar. Okay? So that's going to be important as we move on to understand the photosynthetic reactions, which are not part of this first part of the podcast. I'll pick you up there in the next part of the podcast. Okay? So thank you for watching, and I will see you next time.